Welcome back to the course. So today we'll discuss about co-stimulatory molecule, and this is our lecture number 21. So before co-stimulation, let's try to recapitulate and see what we have understood till now in T and B cell activation. So once the antigen loaded MHC is encountering TCR, which is the T cell receptor, or BCR, which is a B cell receptor, then there are multiple different kinds of signaling that happens downstream to individual cell population, and because of this downstream signaling, the molecules are getting activated. Now, these receptor based biology of the antigen loaded peptide from the antigen presenting cell and the T cell and B cell requires some other molecules in addition to the TCR or BCR. That means in addition to the actual receptor interaction between the antigen presenting cell and the B and T cell, we also need to have some other participation of the proteins which are expressed either by the antigen presenting cell or the T cell or B cell, majorly T cell. Those all proteins are actually important, and these are making a part of co-stimulation. So these are also called as co-stimulatory molecules. So we will try to understand in this lecture about all these co-stimulation, how this could lead to generation of activated T cell immune response, or in some cases how it can help to proliferate the T cell population and do some kind of an immune stimulation in the cell types. So let's try to see the co-stimulation phenomena. So co-stimulation is the essential secondary signal which is required for the immune cell, particularly the T cell population, to achieve the full activation, proliferation and differentiation, preventing anergy or apoptosis. So each and every term is specific here. So what's there is these are particularly important for the T cell, although we do have co-stimulation in the B cell as well, but generally when we are talking about co-stimulation, it's mostly from the T cell point of view. It requires, in order to give a full activation of the T cell, so unless and until you can't say that the T cell are fully activated unless until you have a co-stimulation. It requires for proliferation as well. So once there is a cell which gets completely activated, then it requires to be more in numbers, so proliferations are also required. It further getting into differentiated so that it can do individual function, and it also ultimately leading to avoiding say prevent apoptosis phenomena, so that the cells can survive and do the function. Otherwise, if the cells are not co-stimulated in a proper way, then the cells will undergo either anergy, which is functional inactivation, or apoptosis, which is programmed cell death. So there are proteins which are involved in this process, and these are present in either T cell or in the antigen presenting cell. So some of the molecules called CD28, again these are the cluster of differentiation proteins which are present on the cell, and this acts as a marker. So one such protein is CD28, which is present in T cell population or T cell surface. And then again, and some of the ligands which are present on the antigen presenting cells are CD80 or 86. These are also called as B7-1 or B7-2. So these are again proteins which are expressed in antigen presenting cell, and then those are actually involved in co-stimulation, and once they are expressed by the particular cell population, then we see that there is a co-stimulation happening. Other key molecules which are listed for this phenomena of co-stimulation in T cell are CD40 and CD40 ligands. So basically one particular cell will express CD40, and to dog those protein, again we need a ligand. So these CD40 and 40 ligands are cognate protein to each other. Then apart from that, we have ICOS, OX40, 4-1BB. These are all protein which are particularly involved in memory development. So some of the cells which are involved in memory immune response are requiring those kind of co-stimulation. So we could see that there are various specific cells, cellular protein which are involved in activation process, or there are certain proteins which are responsible for memory cell regulation, and these are all important because this will help in differentiation of the cell type to a particular function. And again, we will see it a little later, but just for the knowledge of understanding this co-stimulation can also be inhibitory functionality. Say they can also do a checkpoint aspect, so they can inhibit the T cell proliferation if the co-stimulation goes in a wrong direction. So some of the receptors such as CTLA4 and PD1 which act as a blocker of the co-stimulatory pathway. So we can also call it co-inhibitory pathways or checkpoint inhibitors. So we'll see later about CTLA4 and PD1 in details, and we will also emphasize the role of CTLA4 and PD1 blockage in cancer immunotherapy. Right? How people are trying to look into the cancer immunotherapy is through binding of any antibody very specific to CTLA4 or PD1. So we'll see it gradually. So there are different members in this co-stimulatory pathways, and we'll try to look into these molecules towards the point of antigen presenting cells as well as towards the point of T cell receptor. So some of the cells which are expressed on, say, dendritic cell and macrophages are B7-1, which is also called as CD80, and B7-2, which is also called as CD86. There are other molecules such as CD275, which is also called the ICOS ligand, which is present on the dendritic cell, macrophages, B cell, and many other cell types. In addition to that, we have PDL1 ligand and PDL2 ligand, which are basically binding to the PD1 domain in the T cell. So these are also expressed in dendritic cell, macrophages, B cell, endothelial lining cell, and many other tumor cell population also. So these are some of the cells which are expressed in antigen presenting cell, which are presenting the cells to, or presenting the antigen to immune cells such as T cell. While opposite side, when we see that the T cell population, then we can see that there are CD28 which are expressed very specifically by the T cell which are not activated. We call it naive T cell. In addition to that, we have discussed that there are two inhibitory receptors which are causing a blockage of the T cell activation. One is called CTLA4 and the other is called PD1, which is actually present in the cell type which are regulating the tumorous growth, particularly in the tumor cells. These are well explored in terms of, so these CTLA4 and PD1 is actually the proteins which are involved mostly towards the inhibition of T cell activation and leading to, say, tumor suppression or immunosuppression condition in case of cancer patients. In addition to that, we have ICOS, which is actually another protein which are expressed by the T cell, and these are generated on the follicular T follicular helper cell. So essentially what we have seen that these particular ligands which are on the antigen presenting cell can bind to this receptor on the T cell. These are apart from this receptor which are present on T and B cell, where MHC1 or MHC2 expression on the T lymphocyte can, so these are apart from the MHC1 and TCR or BCR receptor biology. So there are a lot of variable and constant domain, exactly like the immunoglobulin chain. So these are all immunoglobulin family members. So we can see that these are cytoplasmic tail and the amino terminal region of a protein. There are regions which are variable domain and then a constant domain. Same thing goes here, where we have a constant domain and then we have transmembrane domain. And of course, we have the region that can bind to the separate in opposite direction with different protein is one which are variable in nature. So CTLA4 is what we have discussed, cytotoxic T lymphocyte antigen 4, and PD1 is programmed cell death protein 1, while ICOS is inducible co-stimulator. So the full form of the protein are listed here just for your reference, but otherwise the main take home message from this slide is there are proteins which are expressed on antigen presenting cell, there are proteins which are expressed on T cell, and these are in addition to the one which is already documented or with the one which we already know. So this can help in co-stimulation of the cell types. So let's move on and see how this co-stimulatory molecule works in a very synchronized way in order to stimulate the T cell proliferation and activation of cytokine secretion in the cell. So first of all, so these are the dual dendritic cell, and we understood that these dendritic cells are having a protrusion which are coming because of the actin polymerization. We have looked into the last class that once the protein is getting polymerized, such as the cytoskeleton protein, beta actin or any other actin molecule, then we can see that these are giving flexibility to the cell. Now these dendritic cells are having very high divergence or very high say surface area in order to capture the antigen and all. So once there is an expression of MHC loaded peptide, and once there is a TCR which are expressed on the T cell, once they are bind, then there is an expression of CD28 from the T cell side, and opposite to that CD28 there is an expression of CD40 from the antigen presenting cell. So dendritic cell represent the antigen presenting cell, and this is what is T cell. So these are the molecules which are expressed in a different cell type. So we have to remember this one very critically. We should not confuse that CD40 is expressed in the T cell and CD28 is expressed in dendritic cell. So CD28 is very specific molecule from the T cell side, while CD40 is very specific molecule from the antigen presenting site, which is dendritic cell. To dock with the CD40, we see that CD40 ligand is expressed from the T cell. So CD40 ligand is expressed from the T cell in order to dock the CD40. So this is what the co-stimulation stands for. There is another expression of the protein. Once there is the docking of CD40 and 40 ligand, then we can see that to cover up CD28 also there is an expression of B7 molecule, which is also called the CD8086, which we have discussed as a co-stimulatory molecule. Now this B7 and CD28 can bind very firmly, and once there is a binding, then we can see that the T cell become active and started proliferation. This can also secrete multiple cytokines, and some of the cytokines are such as IL-2, which is autocrine function. They can bind on the self receptor and then activate a different kind of a pathway altogether into the cell. So we could understand that in addition to the receptor normal TCR and the MHC1 loaded peptide, we require some other molecules to come into picture, and those all molecules are called co-stimulatory molecule, and these activations are important in order to do proliferation of T cell and also to secrete a specific cytokine. So this is very critically the pathway related to T cell. Now let's move on and try to see what else is there in this co-stimulation. So just to summarize the same thing, we have this antigen presenting cell which we call dendritic cell. Just to simplify it and reiterate it in a similar in a different way, that we have T cell and the dendritic cell, and the first signal comes from the TCR, which is a T cell receptor, and the MHC1, while the second signal is coming from co-stimulatory molecule, which is either CD8086 from the antigen presenting cell and CD28 from the side of T cell population. So this is actually a typical stage when T cell getting activated. Now there could be a phenomena when there is a T cell inactivation also. So let's try to understand what is inactivation after understanding this activation stage. So let's see this co-stimulatory pathway in terms of understanding the inhibitory aspect. So say let's do the same thing. When you have an antigen presenting cell, here we can see that the MHC loaded peptide can bind to TCR, and this is the T cell. Once there is a co-stimulatory molecule such as CD28 and B7 comes into picture, then this T cell gets activated. Now once this T cell is getting activated, and if at all there is no requirement of activation of this cell type, we need to understand that there could be some kind of an strong inhibitory or checkpoint protein must be expressed from the cell type, and one such protein is called CTLF4. Now CTLF4 is a very specific protein. In addition, in addition to binding to CD28, B7 can bind to CTLF4. That's happening in this case. So CD28 is docked apart. It's not involved in participating any kind of an binding. So CD28 is just out, and the CTLF4 is binding to the B7 as a co-stimulatory pathway. And if this is happening, then we can see that there is a T cell inhibition. So this CTLF4 is a very important molecule which takes part in inhibitory function. So in 2018, the Nobel Prize in Physiology and Medicine is jointly given to James Allison and Dr. Honjo, and these two people independently showed that these two molecules are very important: CTLF4, which is involved in cancer immunotherapy, and also PD1, which is what we'll see a little later, that how they are regulating the treatment of cancer when we can target very specifically CD4 and PD1 molecules. Now, as I said that these are very important in T cell inhibitory pathway, we can see that the cancer cell behaves very differently. When it comes to cancer cell, there are a lot of molecules which are getting upregulated or downregulated. Very critically, the immune cells are also very importantly affected in cancer patient. So generally there is a immunosuppression in cancer patients. So in cancer, we know that the cells are getting uncontrolled proliferation, and the body immune system doesn't recognize it as a foreign molecule because it's all self cell. The self cell or our own body cell behaves differently and start proliferating in uncontrolled way, so that to produce a tumor mass and all. So if there is an inhibition, then we could understand that the T cell won't proliferate, and if there is no proliferation, then probably the cancer patients are not having any kind of immune response that can help them to fight against the extra cells which are produced in the body. In order to do that, we need to make this particular cell which got inhibited by this CTLF4 and B7 co-stimulatory biology, we need to make the T cell active. So what we could do is we can design a CTLF4 specific antibody, so that the antibody against CTLF4 can bind to this CTLF4, and therefore the CTLF4 is not available freely to bind to B7, and therefore now CD28 can bind to B7 molecule, and therefore this T cell remains active in terms of doing its functionality, so that it can go and target the tumor cells. So this phenomena of reactivating CD28 molecule binding to CTLF4 by an antibody is a promising therapy for cancer treatment. So this is what we will try to discuss in other slides as well, but for time being we should understand that in the way the T cell is proliferated, when there is a CD40/40 ligand expression, CD28 and B7 expression, there is an inhibition when CD28 B7 expression, CD28 and B7 attachment is not happening in a proper way, and instead of 28 the B7 can bind to CTLF4, then there could be an inhibition. To curtail this inhibition, to stop this inhibition, we can bypass this process by spatially binding any foreign antibody to CTLF4, so that the CTLF4 can be go out and B7 and 28 can bind to start the proliferation of T cell population. So this is what is the inhibitory pathway that's very important in this co-stimulatory biology. So this antigen presenting cell is what we have understood in T cell activation pathway. Again, the signal 1 is the same which we have discussed in the activation pathway as well, that MHC loaded peptide can dock to the TCR complex present in the T cell, while in the signal 2, instead of CD28 we are seeing here CTLF4. So once there is an signal 2 associated with CTLA4, we can see that the T cells are getting down-regulated. The T cell is not getting activated. There could be an inhibition of the T cell population. And therefore, we need to curtail this CTLA4 expression in order to make the T cell functional for a longer duration of time. So let's try to understand, since this biology has explored a lot towards the cancer treatment. So what's happening in the cancer treatment is a little different, because we discussed that the cancer cell behave differently. A lot of proteins are getting upregulated, many proteins are getting downregulated in cancer cell, and there could be a lot of phenomena that's happening in the cell which could perturb the immune response, that the immune cells are not recognizing these cells as a foreign cell or cells which are deviated from the normal cell population. So let's see some examples here. So what is happening in the tumor cell? So the tumor cell is expressing some kind of a protein such as TRA, which is tumor rejection antigen. These are expressed by MHC class 1, but we could see that there is a loss of CD80 expression. So if there is loss of CD80 expression in the antigen presenting cell, we could understand that there could not be any binding to CD28, because CD28 can dock only to CD80 or 86, which is also called a B7 molecule. So we can see that these are interchanged used in the slides here, but just keep in mind that CD80 is what is B7-1 and CD86 is what is B7-2. So, so 1 and 2, B7-1 and 2 is nothing but CD80 and 86. So once there is a downregulation of CD80 molecules from the tumor cell, we won't see that there is no cognate protein that is available for CD28 binding, and therefore if there is no activation, this T cell is not able to proliferate, and there is no suppression of the tumor cell. And tumor cell, if not suppressed in a proper way, then there could be a progression of more number of cells. There is more number of mitosis that could happen, and there could be more proliferation of cell types. So we can look into a graph here. So if this is x-axis where there is a time and y-axis there is a tumor size, we could see that when there is a small mass of tumor, if there is a exhaustion of the T cell because of this biology of CD28 not docking to CD80, since the tumor cell won't express CD80, there is no co-stimulation happening in this case, and therefore there is no T cell population. The cell will proliferate hugely, the cell may become metastatic, and the large number of cell proliferation could lead to death of an individual in cancer. So tumor immunosuppression in the tumor cases, or particularly in the cancer patient, is the very important area of research nowadays, where people are trying to enhance the immune response towards the cancer cell, because in general the environment is immunosuppressive in nature when there is a proliferation of cell type. And this is what we discussed, that can we do some kind of a CTLA4 and PD1 specific biology in order to channelize the T cell towards the tumor mass, and so that you can still clear the tumor cell in a proper way. So again the same picture which says about the CTLA4 and PD1 signaling in tumor immunotherapy. So let's try to understand it in a more detailed way, although we know that what is happening here when we can block the CTLA and PD1 molecules for enhancing the T cell population. So what is happening in the lymph node, that we have a dendritic cell, and dendritic cell can bind to the T cell receptor, and because of this receptor we can see that MHC1 molecules loaded with the peptide can bind to TCR, so this is an activation signal. CD28 and B7 is a co-stimulation, and once there is a co-stimulation we can see that there could be some proliferation happening. And of course in the naive T cell, when there is an expression of CTLA4, we could block this CTLA4 by antibody, and therefore this CTLA4 is not available to bind to this B7, which is CD80. So essentially what is happening here, that the B7 molecule expression is there in the dendritic cell or antigen presenting cell, but this attachment of P7 molecule from CTLA4 is aborted by the addition of this antibody. So this antibody could be a therapeutic antibody which can block CTLA4, and therefore this normal activation process can go, and this T cell, the proliferated T cell which are coming from the lymph node, can go to the peripheral area. And what will happen in the peripheral area is the tumor cells are there, so effectively they can go and bind to the tumor cell area. Now again there is problem because the tumor cell can have expression of PD-1 ligand. So essentially we can design some of the antibody which can bind to either PD-L1, which is a PD-1 ligand, or we can design some antibody which can bind to PD-1. So essentially the activated T cell can produce this protein which can bind to the PD-1, and again this can block the inhibitory pathway. So this can block the T cell signal in order to proliferate, and to stop this proliferation we can use some antibody against PD-1. So what we could understand here, that there are two ways control. One is towards the central side, that means in the lymphoid organ, and the other control is towards the peripheral organ or peripheral tissue. So in the central we have to block the expression of or the binding of CTLA4 to the B7 molecule by an antibody, and the same way in the effector phase, when there is a peripheral tissue, we need to bind some of the antibody which can block the PD-1 binding to PD-1 ligand, which is basically important for say inhibition of the T cell proliferation in the cancerous cell. So this is in a nutshell about the tumor immunotherapy, which is very rich area of research nowadays, where people are trying to understand this biology in more details, what is the use of CTLA4 and PD-1 in context of tumor immunotherapy. So let's try to understand the final stage of signal integration. So when we are saying, so this is just a pictorial representation which you can understand in terms of say signaling a protein with the ligand. So say there could be a possibility, so this is one condition where there could be shared ligand pathway. So what is happening, the ligands are common for these two receptors. So these receptor 1 and 2 can bind to the same kind of a ligand, and that could happen in case of many different types of cancerous cell, or in the tumor cell, or any kind of other immune cells, where the receptor, the protein which are present on the cell surface, can bind to multiple ligands. And this is very common in viruses also. For example, suppose these are these are viruses, and in the cellular surfaces there are many receptors such as sialic acid receptor. Some other receptors are also present. So viruses always dodge the receptor biology. They can bind to one receptor one time, and maybe they can utilize some other receptor in other cell type. For example, some viruses can use sialic acid more in one cell type, while if they infect the neuronal cell, they can use some other receptor more frequently as compared to sialic acid. So the viruses can do very frequently this biology, and similarly this is happening in the cellular system as well, that we have a common ligand, and this ligand can be bind to two different receptors, and this receptor can pass on the signal to do an effector function, which is a transcription of a gene differently. And similarly we can see that they can use to proliferate and do function differently, although they are sharing the same kind of a ligand, but their functions are entirely different. Second, there could be ways where there could be a shared signaling. So shared signaling stands for, say for example, there are different ligands altogether. So these two are different ligands, they can bind to the receptor differently. But once they are activated in the step 1, the receptor binding, their signal transduction is common. So they can go through the same kind of a signaling pathway to activate the transcription of a gene. That means there could be a way when the signals are shared. So signaling pathways are common, while in the first case the ligand was common. Again, there are many cell types which can do in this way. So we need to understand that how these signals are getting integrated when it comes to immune cell biology, and there are multiple receptors of it. This is just to conceptually understand that how this signal and ligand biology works in order to do an effector function. And effector function here is particularly the production of a protein, or transcription of a gene to produce an effector molecule such as protein. The next one is shared effector, Now effector, shared effector is the last product, which is a transcription. So we can see that the separate ligand can bind to separate receptor, can produce a separate kind of signal, and same thing is happening here. But then these two signal transduction can do a similar kind of transcription, so that a same kind of a gene can do an effector function. This could exponentially increase the gene expression, because earlier only one transcription factor can do a gene expression. Now this can happen because of the addition. So there could be an additional effect here, and once this could happen when there is an excessive requirement of some of the proteins in the immune cell. So you require some of the shared effector effect when there is an excessive requirement of the cytokine or some of the gene required for T cell activation. So these are some of the key mechanisms of signal integration. Let's see and understand few more details when there is a cross activation. So what is happening in the cross activation is, suppose these are ligand which are binding to receptor and doing some kind of signal transduction. We have an effector here. So some of the proteins which are produced here can start a separate signaling cascade. So these are conditional effect actually that can happen once there is a production, while there could be non-conditional effect when the signal can be cascade in a very different way. So there could be a ligand which can bind to the receptor, can do signal transduction and produce a gene expression. Now maybe the receptor can be utilized to perturb and do a different pathway here. The signal transduction can happen and do some other pathways in unconditional way, and also some of the things can do the other pathway can lead to the gene transcription as well. That means these two phenomena, although these are coming from a different ligand altogether, this could be mixed with each other differently. So these are non-conditional, but here the things are conditional. Once there is a production of a protein, then only this get activated. So these are conditional effect, while there could be other phenomena which could be non-conditional. So we could understand that there are many ways the signals are integrated immune cells in order to do a functionality. So there could be cross inhibition, which we have understood in CTLA4 and PD1 expression, that suppose ligand is binding, a receptor is docked to this ligand, we can see the signal, but once receptor is bind to the ligand, it can do an inhibitory effect of the different molecule altogether. So we have a separate molecule ligand which can bind to the receptor and can do a signal transduction. We can see that some of the other protein can do inhibition of the signal transduction, and of course once there is a signal transduction, we don't see the protein production of this one, but we can see the protein production of this pathway effectively. Although some of the signals are diluted out, so the production will be lesser as compared to the original one, but of course there could be an expression when there is a cross inhibition from one molecule to the other one. Now we could understand that these biological processes are very complex. One can do inhibition and one can do activation at the same time based on the physiological condition requirement and the expression of the cytokine and stuff like that. Now we can just finish the lecture with by giving an example. For example, suppose this is a cell which is doing a transcription. Suppose this is a DNA which is doing a transcription of an RNA by DNA-dependent RNA polymerase. This RNA is going into cytoplasm, binding to ribosome to produce a protein by a process called translation. So this is translation and this is transcription. So essentially one can understand that these processes are going on. Now suppose if there are more messenger RNA present in the cytoplasm, what could be the interpretation? There are many ways people can interpret the data. One can say that there is a more production of the RNA from the nucleus coming to the cytoplasm. So there is enhanced transcription that's happening. The other person can say, well, transcription is happening, but translation is getting inhibited, and therefore the mRNA getting converted into protein is now reduced, and therefore you could see that more and more messenger RNA are getting accumulated in the cytoplasm, but transcription is all right. That means the same phenomena can be interpreted by two different ways. One can say that there is more uncontrolled transcription happening, and therefore we can see more and more mRNA coming into the cytoplasm. The others can say that, look, transcription is fine, but somehow the protein translation is inhibited in a way so that the mRNA is not utilized in a proper way, and therefore we can see more accumulation of the mRNA into the cytoplasm. Now both explanation is correct. Unless and until you check the concentration of the protein, you can't say anything, right? So therefore the ultimate thing which you need to check is actually the protein levels, the functionality of the stuff, and therefore the whole biological system is controlled in all different way. People in genomics can see only what is happening in the genome level. The people in transcriptomic or mRNA biology can think about all mRNA expression, while the proteomics biology thinks about what is happening to the protein level. And therefore these all signal integration cascade is very complex phenomena. Just for the knowledge, we need to understand that this can be controlled in multiple ways. Ligand can bind to the receptor, two ligand can bind to the same receptor, the receptor can share, the receptor can do an inhibition, receptor can do other kind of stimulation just to make a cellular homeostasis in a proper way. So this is all about the co-stimulatory pathway and the signal integration. We'll try to look into some other phenomena in this chapter in our next class. Thank you.